So we're really in an amazing time right now, right? We've been through this initial enthusiasm for the great effects we're seeing for a minor subset of patients with many diseases. And we've had now several drugs that have not worked anywhere near as well as we thought they might. The IDO inhibitor, the anti-OX40s, the anti-GITR, given those as single agents or in combination, have not given the kind of synergies that everybody or many people were hoping for. But in fact, I would like to suggest that is exactly what the mouse models would have predicted. The models that are very difficult to treat, that are poorly immunogenic, that don't have T-cell infiltrates, those tumors do not respond, in our experience, to anti-OX40 or anti-GITR or anti-CD27. Those therapies to work in those animal models need to have the T-cell triggered because those receptors are not on all the time. Those receptors get upregulated shortly after the T-cell is triggered. So in poorly immunogenic models of mouse tumors, or in human cancers where you have the cold or the non-inflamed tumor environment, you would not expect those agents to impact those tumors because you're not going to have T cells turned on. They can't give the gas to the immune system to boost it up. So what I talked about today was the concept that we need to do something else. And in our models, it's primarily using tumor vaccines, not tumor vaccines with a single antigen, but with hundreds of antigens that are overexpressed by many of the cancers so that you can take advantage of whatever the T-cell repertoire of a patient is. If you give them one or two antigens, they may not have the T-cells that can recognize those. But if you give them hundreds of antigens that are overexpressed by the patient's cancer, there's a much higher likelihood that you'll turn on the immune system to recognize some of those. And I showed that data from a US NCI-funded clinical trial we ran in lung cancer patients that showed we got robust induction of immunity and boosting of immunity in those patients. So now the point is, how can we make that stronger? And that's where we think the T-cell agonists will work. So giving a vaccine to turn on the immune system, giving the agonist then to boost the immune system, that will give us this, what we think is going to be a big increase. That's what the preclinical animal models have showed us. We've seen great responses and published that. Long-term survival, apparent cures in animals with these combinations. So the next step is to translate that into patients, and so hopefully by next year we'll have data in women with advanced triple-negative breast cancer and in head and neck cancer and maybe another cancer where we'll have combined a combination immunotherapy that includes both activation of T cells with very diverse antigens, like the combination of this autophagisome vaccine that we have, with a T cell agonist, an OX40 or an anti-GITR, and then with some time, a delay, once we've turned on the immune system, we're going to add in anti-PD-1 because of some of the things you've heard about sequence and how important that is, that we believe that too based on our animal models. But it's not just going to be on our belief, it's going to be we're going to monitor the patients and assess that the mechanisms we think are operational or should be being turned on are actually being turned on, and that we're adding the anti-PD-1 or the anti-PD-L1 at an appropriate time when it's needed. So I think it's a very bright time, and for many patients to go on clinical trials that are going to take these best and these new combinations and give them a pretty good opportunity, we hope, to impact their life, and working towards this idea that we'll be able to ultimately cure everybody of their cancer.